I would now invite the first speaker. This is George Jen from Tsinghua University in Beijing. I was told that he's working at the interface between microbiology and generation of materials, and accordingly, his presentation will be on applications of synthetic biology in industrial biotechnology. So it's all yours.
Thank you, Mr. Chairman. Taking this opportunity, I would like to thank the organizing committee for inviting me here, and also the local host that have done an excellent job to offer us such a very comfortable stay and comfortable conferences. Industrial biotechnology is one of the areas that synthetic biology can apply. And my research is mostly focusing on the application of polyhydroxyalkanol and its production. So if we look at the impact of the biotechnology, the synthetic biology to industrial biotechnology, we can see that the microorganism can be exploited for energy production, material, and chemicals, as we had heard from the last 2-day lectures. And we know industrial biotechnology relies on microorganisms for production of energy, material, and chemicals. However, we know for microbiological process to compete with chemical process, you're always limited by the less unique production ability, or we call YEP, or product density. So the biological production rate is much lower compared with the chemical one. So because and also they require a lot of energy input compared with the chemical one. Sometimes the chemical reaction they are running at a very quick rate. And so this slide in Chinese, but I like this slide very much. We are experiencing a very good rapid growth in the what we call the petrochemical industry revolution, but we are heavily loaded with environmental crisis, energy crisis, and resource crisis. Now we're moving to bioeconomy, that is a developing area for what we call industrial biotechnology or industrial microbiology. So construction of pathway that will allow cells to grow at a very higher microbial density is very important. It's almost the key to improve the production yield. So this will be very important. And on the other hand, if we can synthesize pathway that can turn aerobic process into micro or anaerobic process that will reduce energy consumption for the biological process significantly, and that will, of course, reduce the cost for biological process. So what we do here is we try to construct a synthetic pathway that can turn aerobic process to micro or anaerobic process to get higher cell density. And this is very important, as it can dramatically reduce energy for the microbial process. As we know, the aerobic process require intensive mechanical stirring and intensive aeration that will require a lot of energy. Where we know that some of the organisms like Pichia pastoris or Escherichia coli, they are living in a facultative aerobic process. That means they can grow in an aerobic process and in a lower oxygen aeration process, so that they don't need intensive aeration and stirring, magnetic stirring, that will reduce energy, so that we can see the higher cell density in E. coli fermentation or in Pichia fermentation can be achieved conveniently, but you will have difficulty to achieve higher density if a microbial process is aerobic one, because you will run out of oxygen at a higher cell density. So higher cell density is very important for the production of what we call the PHA products. PHA is a microbial process. I will introduce a little bit later. And this product is an intracellular polyester, and that has to be rely on the cell density for efficient recovery. What we do here is we try to make a higher cell density to reduce the downstream cost and energy consumption. And of course, if we succeed, we also want to reduce the production time for improving on the yield of PHA and also reduce energy costs. So, but the issue is how to reduce energy and at the same time achieve higher cell density growth. And this will be depending on how to allow the cell to continuously produce energy during the stationary phases for the cell growth. So this is PHA. What I just mentioned, it is a bioplastics with very diversified structure, and it normally is an inclusion body in the bacterial cell. And as reported in the literature, the PHA can be accumulated up to 96% of the cell dry weight. So this is amazing. And therefore, it's becoming very interesting for the plastic industry that are looking for environmentally friendly packaging and for biodegradation of the plastics. So the molecular structure of this material is very diversified. It has been reported that 150 different molecular structure, monomer structures have been discovered, and these are just a few that has been commonly encountered in the PHA research. And we had something called short-chain length PHA, which has a very short-chain monomer, and some of them had a longer one. So depending on what kind of organism you are using, or depending on what growth condition we employ, we get different molecular structure. In a mixed population, for example, in a microbial population, we normally discover in sewage, in activated sludge, we get a mixture of these structures. I will discuss this, how we can use that. And as I mentioned, that PHA production cost is highly dependent on cell density. The higher the cell density, the lower the production cost. So one of the organisms that we use to produce PHA is Pseudomonas putida. And this is an organism that is strictly aerobic, and they can produce PHA from either fatty acid or from oxygen. But because it's absolutely aerobic, it requires a lot of oxygen for the growth. Especially if you're trying to grow them to higher cell density, you have to really pump in a lot of air into the fermenter, and you have to intensively stir in the fermenter to improve the oxygen transport. And this is very energy consuming, and especially energy is very expensive nowadays. So it's becoming very important to reduce energy consumption by an anaerobic process. As I said, it's difficult to produce higher cell density and PHA production due to the intensity to supply oxygen. So what we're trying to do is, could we turn the process into an energy-saving one? So I said that the material can use fatty acid or glucose. So what we're trying to do, if we can intensify the beta oxidation pathway to generate more reducing power or energy, then we could probably improve the growth and the PHA production. So as I said, the material require intensive aeration for MADH consumption and for energy regeneration. So the key is to reduce dependence on oxygen. So if we can reduce oxygen demand, then we can probably improve the PHA production as a whole. So friction oxygen is required for the oxidation of MADH, and then you get more ATP to power the beta oxidation and for the cell growth. So how to solve the cell density problem? Traditionally, I had just mentioned you have magnetic stirring, more air supply, and this is on the cost of higher energy consumption. Or if we can somehow turn the strictly aerobic microorganism into a facultative microbes, we probably achieve higher cell density. And this has been reported in E. coli, Ralstonia eutropha, Pichia pastoris, which are, as mentioned, facultative aerobic, which means that they grow either both in the aerobic condition and in the anaerobic condition. So we try to do that to achieve this purpose. So if we look at the anaerobic pathway in E. coli, we can see that they are pyruvate to different compounds, and they are pyruvate to acetyl-CoA, acetaldehyde, and many others. All of these produce MADH, and this MADH, of course, can be oxidized to generate ATP. And enzymes involved in this process is quite a few, like PFAB, ADHE, and ADHE, PTA, ACT, and so on. And if we can clone this anaerobic pathway into an aerobic pathway, and probably we can recycle MADH, and thus we hope to improve the PHA production and, of course, cell density by doing this MADH recycle. So if we look at the energy balance under anaerobic condition involving this gene we had discussed, so we had the following pathway that consume the MADH. You can see that we had the acetyl-CoA pathway, and we had the acetyl-CoA, the glucose pathway, and we had the pyruvate pathway, and all these combined together, we are able to turn the MADH into MAD and generate some ATP for the synthesis. And in addition, if we introduce some enzyme called MADH oxidase, that can also help to oxidize MADH and then reduce energy. In that case, we don't need to grow the obligate aerobic organism under oxygen condition. We can grow them in the absence of oxygen by cloning this pathway into the obligate aerobic organisms. So what we do is, as said, we clone the strictly aerobic pathway organism, Pseudomonas, with the acetate pathway, with the ethanol pathway, and with the lactate pathway, and with MADH oxidase. All these can help conceal NADH and then turn this MADH reducing power to ATP, and thus we are able to power the growth of microorganisms and, of course, accumulation of PHA. And then we got probably a facultative aerobic Pseudomonas strain, and that can grow to higher cell density. This is what we're trying to achieve. And besides this pathway, the NADH oxidase and some anaerobic promoter should be cloned into the gene so that the process can be really run in anaerobic conditions. So this, if succeeded, could lead to a reduction on energy consumption and improved cell density and thus PHA production, and hopefully making the process more compatible with the chemical ones. So these are enzymes that we're interested to clone because they are involved in anaerobic growth, or at least for Escherichia coli, and they are the key to recycle NADH. So we had succeeded in cloning all these genes, and these genes are turning into the biobricks using the synthetic biology method we discussed in the last few days. So we make the biobricks and then construct the what we call the gene cluster, and we clone them into the organism, and like this. And of course, we had to clone the PHA synthesis gene together in a separate plasmid. And all of these are turning into the organism, and then we can see that under this is a wild type, under the aerobic condition, we had a higher pH content, and we also had a higher cell growth rate. But under the absence of oxygen, normally the growth is very poor. However, if we clone the pathway into the organism, and we also replace the promoter with an anaerobic promoter, alcohol dehydrogenase gene promoter, then together with the PHA synthesis gene, and of course combined with the anaerobic gene involved in E. coli, then we were able to produce the PHA from a lower intensity of 29% cell dry weight to content of about 48% under the action of anaerobic promoter. And this means that the cloning of all these could be very successful. However, this work is still ongoing, and we hope to improve the cell density further and improve the PHA production further by expressing more the anaerobic process related genes. This is one of the conclusion we can make. It's possible to construct synthesis pathway that turn aerobic process to a micro or anaerobic process. Thus, we can improve the growth of the cell, the growth of cell to higher density. And of course, PHA production can be improved from 29% to 48% of dry weight under anaerobic conditions. But this is one way to reduce energy consumption. And for the PHA production, we also need to have a higher substrate to product transformation yield. For doing this, if we look at the central pathway, the TCA pathway, we can see that the TCA pathway actually lost some oxygen. And normally the TCA, some CO2, the TCA cycle is running in this way. If we can turn the TCA cycle into an anaerobic one, say that turning the PCA around, maybe we can save some energy consumption. And this is what we call the reverse TCA cycle that can help maintain some CO2 in the circles. What is reverse TCA? We know that some organisms, especially some archaea, like this one, is more protos, neutrophils. And this is a phototrophic organism that grows under anaerobic conditions. And it has a reverse TCA cycle. If the TCA is running in a clockwise way, this is what we call the TCA, normal TCA. If the TCA is running the other way, then we call it a reverse TCA. If you see this picture, the TCA, if running this way, we had a CO2 release. If we run it the other way, then we can recover 2 CO2 in the process. So if we look at this theory, for PHA production via the TCA cycle, the normal TCA cycle, we have substrate to product yield of 48%, based on this calculation. If we had running the other way, then we had improved yield, which we will discuss later on. And as I said, that we are trying to turn the aerobic process into an anaerobic one. And besides the cloning of the anaerobic process into the aerobic one, we had to recover more CO2 during the production process so that we had a higher substrate to a product transformation efficiency. What we do is trying to improve the utilization of, for example, glucose or fatty acid. There's many possibilities. One of them is to recover CO2 and consume residual NADH in the absence of oxygen, as I just mentioned. And to recover more CO2, the TCA cycle could be constructed also to consume redundant NADH and to recycle CO2. If we look at the pathway of or reverse TCA balance, we would produce 1 phv from glucose. If we're doing in the normal way, if we're doing both, then we will have probably produce 2 phv from 1.5 glucose. And this will be more efficient than the traditional one, TCA cycle. With the reverse TCA, we can improve the cycle from 48 to 64% substrate to product yield. So the first one is to construct a TCA cycle, but it's also equally important to delete the pathway that is not related to PHA production, such as the deletion of mixed acid fermentation. So these are the mixed acid fermentation that consume a lot of substrate, and they produce substrate that is produce products that are unrelated to our final product, polymers. So, and then if you look at the TCA cycle and the mixed acid fermentation, so we had so many substrates to use. And in the reverse TCA cycle, if we run it around, we can produce succinate. Sorry, this is a Chinese succinate from the PEP cycle that can supplement the loss of succinate in the reverse TCA cycle. So we delete several pathways related to the consumption of PHA precursor. and then that will help to channel more substrate towards our polymer production. And these are some of the genes that need to be expressed to run to reverse the TCA cycle. And there are 1, 2, 3, 4, 5 genes, and of course this is for the polymer production. Totally, we have 11 genes to be constructed in the organism to reverse the TCA. I say that the PHA is coming from the glucose through acetyl-CoA to the polymer. If you're running this way, and we had a ATP production that is much better than the normal anaerobic process, and we can achieve a production yield of 48% if this is aerobic one, which means that a normal TCA will generate 48% of the substrate to product yield. So if the way is running in reverse, then we get a 64%, as I mentioned, based on the calculation we have done here. And what we do is to just say that we bring this 11 genes related to the reverse TCA cycle, and then we bring them into a BioBreak structure, and then link them with isocal, this is a difficult name, isocal donor, which will allow us to bring them together in a gene cassette. And finally, if we construct this in the gene or E. coli or whatever pseudomonas, then we will be able to run the anaerobic process. I think my time is about to come, so I will go quickly. So these are genes related to the normal process. If we construct a pathway that can run in the reverse way, then we will turn it around. So pathway that are normally running only in this direction are catalyzed the gene called GLTA. So now we're trying to do is replace that gene that can run the other way, so that we can control the process to go into a reverse way. So we need the gene I just discussed, about 11 genes to run the TCA on the other ways. So if TCA is running in the other way, we have CO2 fixation that will improve the substrate to product yield. And then if that is succeeded, we can use PHA for other purposes, like PHA as biofuel. We have discussed in the last few lectures that PHA, that we have many biofuel. We know that we had a long chain alcohol, we had ethanol, we had biodiesel or hydrogen. However, PHA is also one of the biofuel that we pay attention to, because now we have technology to improve the yield of substrate to PHA production. We might consider that this is also a very useful energy sources because it's a polyester. And bacteria also use this polymer to make energy and cell and carbon source. Why can we not use it for energy purpose? And if we hydrolyze this structure into a methyl ester, and then we generate a whole series of different substrates, and they are very useful. The substrate is quite similar to a biodiesel. We know mostly the fatty acid methyl ester. If we can turn it around, then maybe we can see they are actually combustible. So under the combustion heat study conducted just recently, we know the 3-hydroxybutyl methyl ester has a combustion heat of about 20 kilojoule per gram, and compare with ethanol, which is 27. And these are quite similar to 3-hydroxyalkane methyl ester, quite similar to ethanol, of course lower than diesel and lower than others. But this one has some PHA-based biodiesel has some advantage which we can discuss. The combustion heat is about the same as ethanol, but in an ecosystem, these materials are always produced. This is very important. So if you are trying to find this PHA, you can find the activated sludge, which does not occupy any land. It does not require any substrate input, and it does not need to consume food for producing the biofuel. And also, the PHA production can occupy over 90% of the cells right away. And this is important. As now, the algae can produce about 50% of the fatty acid, but this is not way comparable to PHA production that can be, you can see that, occupy almost the entire cell spaces. So the PHA biofuel has a combustion field or about the same as ethanol. So we have done a mixed blending study combining different PHA-based biofuel with ethanol, diesel, and gasoline. And you can see that they had reduced a little bit of the combustion heat of this traditional traditional fuel, but it's acceptable because these are still combustible. And we also conclude that this blend created a reasonable combustion heat that could be used for a motor. And also these PHA-based biofuel are blended with some new long-chain alcohol like propanol and butanol, and they also generate some reasonable combustion heat that could be used for motors. So we have not done further study yet, but this is very important data that could guide us into the PHA-based biofuel study. So the one thing very important is the cost of PHA biofuel. And to make PHA as a bioplastic is very costly. But to be used as biofuel, and they could be cost competitive. So how to get this cheaper PHA? We can get it from the what we call the waste treatment plant, where a lot of sewage is produced. And this sewage can be guided or manipulated to contain a lot of PHA. We know the sewage is nothing more than microorganisms that grow to higher cell density. So it can also be obtained from some of the sludge that is obtained from the anaerobic production process. And of course, aerobic process produce a lot of activated sludge. Excess sludge also contain a lot of microorganisms. And we have designed a process that can easily recover the PHA-based biofuel from the sludge and then get the right biofuel from the process. Now, this has been tested in a bigger scale, and we hope that it could be a supplement to the existing biofuel process. And we have done a calculation, and we come to conclude that the PHA-based biofuel is about this price, about $1,200 per ton, compared with biodiesel, which is about this prices. This is the Chinese price. I don't know international one, but you can see that PHA-based biofuel is a lot more cheaper. And of course, the combustion heat is not as good. Combustion heat is not as good as biodiesel, but the cost is okay. If you blend it with the other fuel, that would probably reduce the cost further. And I said that we can mix the PHA production substrate with the cell and to improve the PHA production, or we can manipulate the carbon source nitrogen in the growth medium and even in the activated sludge, so we can improve the PHA production. And of course, very important is that it does not use stuff for food and arable lands for making this PHA biofuel. It comes from activated sludge anyway, and activated sludge is produced every year. In China, we had about 10 million tons of dry activated sludge every year, and this, if managed properly, we can use to make biofuel. So it could be cost competitive, and we think it will have a bright future. And my time has come to an end. I would like to thank many of the students contribute to this study. So these are all the students, not all of them are currently, but we come together for a meeting, and they helped develop this technology. And also, I would like to thank Mr. Li Kaishen. He's a very well-known man in Hong Kong, and he supported our what we call the multidisciplinary center, allowing us to combine the chemical expertise, polymer expertise, microbiology, and molecular biological knowledge to build the PHA-based combined study. And thank you for your attention.
Thank you very much for this concise presentation. We have time for one or two short questions.
Okay. The idea of the reverse TCA is very nice, and I guess with molecular biology tools you may be able to do it. But from the thermodynamic point of view, I'm not sure if you can really reverse it, because from thermodynamic point of view you may need energy, right? and how the cell will get the necessary energy for that. That's maybe my first question. And probably the second one is the previous approach to get the ethanol or acetate or lactic acid pathway into Pseudomonas putida. And normally these products are very toxic for growth, right? I understand your goal is to have higher cell density, but this actually very nice, simple process engine point approach to get very high cell density in very short time. At least with E. coli, we can reach more than 140 grams per liter dry biomass within 24 hours. So I'm not sure if you have considered also the process point approach. For the first question, bioenergetics.
Yeah, you're right. Energy is very important in this regard. If you're running this in an anaerobic condition, probably energy consumption is also minimum, I hope. And some of the enzyme actually were used here under anaerobic condition, and we had done a thorough calculation of energy balance if you say under the RTCS cycle, and at least theoretically it is possible to balance the energy in the whole process. But I said that we had some way to go to really achieve this RTCS. But initial results show that the growth actually was slowed down. Maybe you can achieve higher cell density, but the growth is slowed down due to what you said, energy supply problem. With aerobic process, energy supply is not a problem, but with anaerobic one, it's an issue. So growth is much longer compared with the aerobic process due to energy shortage. But under anaerobic condition, anyway, it doesn't consume a lot of energy. So maybe time is not a factor. Thank you.